Um, Leading up to our discussion of blood vessel disorders, namely atherosclerosis of medium to large sized arteries in our body, we really need to talk first about hyperlipidemia, because hyperlipidemia is a major risk factor, one of the strongest risk factors for the development of atherosclerosis. So in order to understand how hyperlipidemia relates to atherosclerosis, we have to kind of talk about how the body handles lipids in our body, and especially cholesterol. So there are three types of lipids found in the body. We've got triglycerides, phospholipids, and we have cholesterol. So because triglycerides and cholesterol are insoluble in plasma, they are encapsulated and travel through the bloodstream by special fat-carrying proteins called lipoproteins. And lipoproteins, as we'll see in just a moment, are really elaborate packages that allow us to carry these types of lipids through the body. So there are 5 types of lipoproteins. Before we take a look at what a lipoprotein looks like, let's just mention what these 5 types are. First, we've got what are called chylomicrons. We have very low density lipoproteins, intermediate density lipoproteins, low density and high density lipoproteins. And so as you can see, the word density is in a lot of these is in the name of a lot of these lipoproteins because they are classified by their density, and we'll see exactly what density really was referring to. So before we look at this slide, let's go forward for a second and look at what the structure of a generic lipoprotein really is. So you'll see on the shell of this lipoprotein, of this package, is a layer of phospholipids. And we know that phospholipids are a great way to travel through the bloodstream because they have a hydrophobic or water-soluble head and fat-soluble tails. So the water-soluble heads face the plasma, and the fatty acid tails are facing the interior of this lipoprotein core. And in the center of this lipoprotein, we have lots of triglycerides, we've got lots of cholesterol. Okay? So those are our 3 basic components of a lipoprotein: phospholipids, triglycerides, and cholesterol. Also on the surface of lipoproteins are proteins called apoproteins. These apoproteins, and there are several different types of apoproteins, act as identifying markers, okay, for these lipoproteins. So each of these 5 different lipoproteins tend to express a different kind of apoprotein. And when a cell interacts with one of these lipoproteins, the apoprotein allows the cell to recognize which one it is, and it kind of determines or directs the metabolic fate of this lipoprotein. So if we look at how these different lipoproteins stack up in terms of density. So basically, if we take a test tube full of plasma and spin it at very high speeds in a centrifuge, what happens is everything that is highest in density tends to get pushed down to the bottom of the test tube, and everything that is lowest in density tends to be at the top. Now, we know that these lipoproteins are made up of 3 different things: triglycerides, cholesterol, and protein. Now, you'll notice that we've got different types of lipoproteins on the left here, on the right here, based on density. And what gives a lipoprotein greater density is the protein content. Okay, so the more protein that is integrated into the structure of the lipoprotein, the higher in density this lipoprotein will be. Okay? And so you see chylomicrons are the least dense, they have very little protein, and they are really mostly triglycerides. Okay? The VLDLs, very low density, they have a little more protein, and they have a little more cholesterol compared to the chylomicrons, but still mostly triglycerides. And then we get into the LDL and the HDL, the low density and the high density. So LDLs, as you can see, are mostly cholesterol. Okay? Some protein, a little more protein than the VLDLs, a little bit of triglyceride. But LDLs are mostly cholesterol, and HDLs are mostly protein. Okay? A little bit of triglyceride, a little bit of cholesterol, mostly protein. Okay? So we're going to talk more about these LDLs versus HDLs, but the purpose of LDLs, okay, LDLs are the lipoproteins that deliver cholesterol to tissue. Okay, so these LDLs are designed to be packages of cholesterol. The chylomicrons, as you can see, they are designed to deliver triglycerides. Okay? And the ones that the lipoproteins that are between chylomicrons and LDLs, the VLDLs or the IDLs, they are simply intermediates. Okay? But on one side of the spectrum, we have chylomicrons that deliver triglycerides, LDLs that deliver cholesterol. And the HDLs are not delivering lipids to cells. In fact, the HDLs, their primary role is to actually actively regulate the amount of LDLs in circulation. Okay. So let's look at how these different lipoproteins relate to one another and how they are used by the body. So starting off here, we have cholesterol and triglycerides that are consumed with our food, and they are absorbed by the intestines. And what the intestines do, the mucosal cells, the epithelial cells that line the interior of the intestines, what they do is they are going to take the triglycerides and they are going to form chylomicrons. And they're going to put those chylomicrons, we're going to absorb dietary cholesterol and triglycerides in the form of chylomicrons. Okay? So when we absorb fat in our food, fat and cholesterol in our food, the unit, the lipoprotein that's formed and enters our bloodstream is the chylomicron. Now the chylomicron, we know, is predominantly triglycerides. So when the chylomicrons get into circulation, they're going to go in a couple of different places. They're going to go to adipose tissue and skeletal muscle. Okay, so some of that triglyceride is going to be stored in their adipose tissue. It's going to be consumed by our skeletal muscle tissue for energy. Okay, so that's one place those chylomicrons go. Another place that the chylomicrons go is to the liver. So the liver takes up lots of chylomicrons and chylomicron fragments, and it's going to disassemble those lipoproteins, and the liver is going to do a couple of different things. The liver is going to produce HDLs, put that into circulation, and the liver is going to also release what we call our very low density lipoproteins, our VLDLs. Okay. Now, the rest of the chylomicrons in circulation, along with the VLDLs produced by the liver, while in circulation, get converted into our intermediate density lipoproteins and then into our low density lipoproteins. Okay. So in circulation, the chylomicron, some of those chylomicrons, along with the VLDLs from the liver, are going to get converted into IDLs and then LDLs. Okay. Now the LDLs are a way to deliver cholesterol to tissue. So why does our body need cholesterol? Number one, cholesterol is part of the plasma membrane. It's a structural lipid. It's part of the plasma membrane for all cells in our body. Okay? So LDLs are going to deliver cholesterol to all tissue in order to maintain plasma membrane structure. Number two, all of the cells that produce steroid hormones, okay, all of our endocrine glands that produce steroid hormones, like our testes, our ovaries, our adrenal cortex, need LDLs, right, as a source of cholesterol in order to produce those steroid hormones. And then finally, our biggest consumer of LDLs is our liver. So our liver takes up something like 70% of the LDLs, okay? And it uses that cholesterol in the LDLs to produce bile. Okay? So those are the three uses for normal uses for LDLs. They're taken up by all cells in our body as a structural lipid, number one. Number two, it's taken up by endocrine glands that produce steroid hormone. And number three, the biggest user of cholesterol is the liver, and it uses that cholesterol to produce bile. Now, this kind of LDL use, okay, is done through what we call receptor-mediated uptake. So these cells, our generic cells, glandular cells, our liver, they have LDL receptors that these LDLs bind to, and then they are taken up that way. And so that is a major route of LDL removal by the body. This is what we call the receptor-dependent pathway of LDL removal. Okay? That is a pathway that we intend LDLs to be taken up by. The second method that LDLs are removed from circulation is by binding to HDLs, and they get returned to the liver. And this is what we call reverse cholesterol transport. HDLs bind to LDLs in circulation, and they come back to the liver. Okay. Now, there is a third way that LDLs get removed from circulation, and this is what we call the scavenger pathway. And this is the pathway that is damaging to the body. You've ever heard of LDLs as being the bad guy cholesterol? It's this pathway that makes it the bad guy cholesterol. Because when we have excess LDLs beyond what's needed for the receptor pathway, beyond what the HDLs can handle, when the remainder of the LDLs are left in circulation, they are going to interact with reactive oxygen species in circulation, because they're always in circulation, and these LDLs become what they call oxidized, okay? Which means they become very reactive and they damage the lining of blood vessels. And when they damage the lining of the blood vessels, you get an inflammatory response, and the LDLs get consumed and removed by macrophages in circulation. Those are the scavenger cells, the macrophages. Okay? So that last way to remove LDLs are after they have damaged the inner lining of the blood vessel, and the macrophages have to come in as part of an inflammatory response, consume them and remove them. Okay? So it's this last scavenger pathway that makes LDLs, when they are too high, and it puts you at increased risk for atherosclerosis. We'll talk about that process later. But this is what makes LDLs sort of the bad guy cholesterol. Okay? And this ability for HDLs to help regulate the level of LDLs is why we call HDLs the good guy. Okay? And so this is why we want someone's LDL level to be as low as possible, and this is why we want their HDL level to be as high as possible. Okay. So when someone has what we call hyperlipidemia or dyslipidemia, this represents an elevation of certain lipoproteins in the bloodstream, whether it's chylomicrons or triglyceride levels, or LDLs, or what have you. Now, the most important thing that we look for in terms of atherosclerotic risk is elevations in cholesterol, so hypercholesterolemia. And hypercholesterolemia the most important thing, so we can look at somebody's total cholesterol, which is the HDLs and the LDLs, but what's more important is to look at the LDLs, okay, because we actually want HDLs to be as high as possible, but we want LDLs to be within below a certain healthy amount. Okay. So when someone's LDLs are abnormally elevated, that really is what represents hypercholesterolemia. And there are really two forms of hypercholesterolemia, primary and secondary. Okay. Now, primary hypercholesterolemia represents elevated cholesterol, namely LDLs, that develop independent of any other health problems or lifestyle behaviors. Okay. So this is not elevated cholesterol because somebody doesn't exercise enough or because they eat foods that are bad for them. Okay. This is a hypercholesterolemia that would be present in this person no matter what they did health-wise. So primary hypercholesterolemia is often genetic in nature. And there are a few identified mechanisms that create this kind of predominantly genetic primary hypercholesterolemia. And that includes, for example, defective synthesis of apoproteins. So the apoproteins on LDLs can't be recognized by cells in our body like the liver, okay? And so they're not taken up by the liver and other cells, and they remain in circulation. Okay. Another example is some sort of problem with the LDL receptor, either low expression or a defective LDL receptors. Okay, so then LDLs can't bind and be taken up by cells, and they remain in circulation. So these are examples of mechanisms that can produce this primary hypercholesterolemia. Much more common is the secondary hypercholesterolemia, which is associated with lifestyle and behavior, and therefore can be controlled through lifestyle and behavior change. So this is some of the lifestyle and behavior risks for secondary hypercholesterolemia includes things like obesity, high caloric intake chronically, diabetes mellitus, and so forth. Okay. So it is the foods that we eat, how much and what kind of foods that are producing this elevated amount of LDLs. Okay. And really, no matter what the mechanism for the hypercholesterolemia, primary or secondary, these individuals have a much, much greater risk for atherosclerosis.